Welcome! Welcome to yet another Cornell keynote. I'm your host, Lutz. I'm faculty at Cornell where I teach about AI, data, machine learning, all the good jazz. And as many as you know, I spent a lot of time trying to figure out what AI actually does to us, to our products, to our society, and so on. My personal work is essentially focused on workflows. I built a search engine, or like search and discovery engine. I built fine-tuning models. And if you are interested in building stuff with me, join one of the e-commerce, eCornell workshops on workflows. I have a version for consumers where you learn to build your own AI agents, and I have a version for enterprise agents where we talk a little bit about scale and that vibe coding is not the end of the story. Now, when we talk AI, very often people talk about chatbots, copilot, apps, vibe coding. But that's not the full story. AI goes way beyond this. It goes beyond a normal chat conversation. We have a physical world. So let's talk about AI in the physical world. Let's talk molecules, materials, medicine. So that's a new frontier. And we can call this BioAI, artificial intelligence applied to biological data, biological problems. Let's move out of the research labs into the real world and let's see what this means. And obviously, the stakes here are high, and we know one of the typical problems to address is cancer. And we have been working for decades on it. So, what will change now when we have better technology to address this? So, with that, I'm very delighted to be joined today by Cyriac Roeding. He's co-founder and CEO from Earli. He is, as you will very quickly figure out, a fellow German. He
Yeah,
has
I'm sorry about that.
like not that German. Don't excuse yourself there for that already. No, but he is the CEO of a bio AI company developing methods where you essentially create little switches or robots inside from cancer cells. It's super fascinating. Now, Cyriac is in his fourth company. He's one of those amazing guys who go through Silicon Valley and create one venture after the other and created a lot of value. He raised by now more than 150 million from investors, including A16Z, Koshla, Perspective, and J&J. He sits on scientific advisory boards, included Nobel laureate Jim Allison and Moderna co-founder Bob Langer. Before Early, Cyriac built Shopkick in Germany and sold it then for 250 million. And therefore, he was, or still is, I think, an early investor in OpenAI. He is an LP at many different funds, and he co-authored the Harvard Business Press book at 25. This is like, I mean, I don't really know how to introduce you otherwise, like in other words, than he is kick-ass. So here he is, Cyriac. So glad to have you here.
All right, it's a pleasure to be here, and thanks for having me. I've heard a lot about this show from you, and so I'm excited to be here today.
So before we go into the typical story flow where we talk about you and who you are, but let's first start with the big news. You closed Series B. Congrats. That's a big step. Like, tell us a little bit about
Oh,
it.
I wish you were right. We haven't closed Series B.
Oh,
We
like,
just
are you
started it.
close? Are you
We
so like
just
so?
we just started fundraising on it. We have raised 105 million for Early so far, and now we have to raise more money because we're going to take this whole thing actually into humans. And for that, you need a little bit of money because clinical trials are not the cheapest thing on the planet. So that's what we're going to do next. And just one thing, the last company you mentioned was Shopkick that was in the US actually.
Oh,
We made it easy for people to get rewarded just for walking into stores physically, into a real store you can touch, which is the topic of today. You walk in, use your smartphone as a digital player on top of the real world, the non-digital world, the physical world, and then you get rewarded just for being physically there in points, and the points turn into gift cards. And that company That company was acquired for 250 million. Kleiner put 7 million in, got 78 million back out. And then I wanted to do something different, but everything I looked at in hindsight, I literally just did what I wanted, looked at what I was excited about. And I came to very different things like
You told
brain.
me about this, right? You kind of like you went around Silicon Valley essentially
Yeah,
trying to figure out what's the next cool thing.
that's right. That's right. It led me to brain machine interfaces, computer interfaces. I literally helped a guy sitting in a dilapidated building in Berkeley shooting microwaves at his own head, trying to decipher his own thoughts. And this was in, yeah, it's true, in 2016. And it was very, very early. I also looked at the other thing that is my passion and my love is robots, consumer robots. And on that intersection, it's obvious you have the physical hardware, the atom, and the digital software, the bit, and the atom meets the bit. And then the third thing I looked at was biology meets AI.
But
And
in
now
all
that's a
three
really
cases,
interesting world. Sorry?
now, no, in all three, like in all cases, also if you compare this with Shopkick before, you always try to figure out how can I get AI workable or how can I get technology workable in real world, right? So
That's
you
exactly
are,
it. I am fascinated with the intersection of the digital bit and the physical atom. Where those two meet, I think you get the real fundamental innovations because we live in the physical world. We live in no other world. We live
yeah,
in the actual touchable world. And when software can start manipulating the actual touchable world, then it becomes very, very interesting and potentially game changing in how we live. So I'm actually much more interested in that than I am in pure software alone. And that's, by the way, a great other analogy is what is more interesting, VR or AR? Virtual reality or augmented reality? What is
augmented, if
more
you
in,
ask me, absolutely
yeah.
augmented.
What's more interesting to you? Is it more interesting to sit with a cap here and you look like a weirdo and you look at your own world and you live in it and you have no interaction with the
The
actual
typical
world
Apple glasses, right? Like,
yeah, it's really awkward. Or are you more interested in glasses that actually put an overlay on the physical world on top of your glasses and you can see information about what you're looking at. You can connect with people, get their backgrounds based on their LinkedIn profile when you talk to them at a conference. What if you can have, while you are working as a surgeon, get information displayed in front of your eye while you are actually operating on the patient to make sure you're cutting all the tumor out? So that's where it becomes very interesting. And that to me is the classic an example of physical meets digital is more interesting than just digital alone.
now, before, and I didn't plan to dig into this, but like if we say physical meets digital, where do you see the big blocks of what's happening today? And like you know Silicon Valley pretty nicely, and so like obviously we will talk cancer for the rest of the show, but are there other blocks? You mentioned the glasses. What else is in your mind when you
Yeah,
say
it's a great topic. So I think in a nutshell, to keep it really brief, I think the big, massive innovations are going to happen, A, in robotics. And I think in 20 years, not having a robot living in your house is going to be as strange as not having a washing machine today.
yeah.
Two, glasses are the next big computing platform. This is the next platform.
Yes, I totally agree. I actually tried very like I was the first. This is the first version. Like, dear Meta, can you please fix those glasses? They are terrible in the user interface. They're still
Hmm.
too clunky, but yeah, like yes, I agree. I need a new pair of glasses. So if you know any good system, let me know.
So, whoever owns this platform is going to own the next big
Yeah, I agree.
substantial platform. We started with the watch, and the watch is very interesting for fitness and for health monitoring. But the glasses are the direct connection to what you're thinking, what you're doing, and it's remarkable. And of course, the next
And
big
they are
move.
always, I mean, like some of people have like the like whatever, like you like the necklace don't will not work so much. Glasses are actually they're always pointed in the direction you are actually looking at,
That's
or
right.
the
And a lot of
necklace or not.
glasses, a lot of people wear glasses anyway. But even those people who don't wear glasses all the time, they usually have sunglasses. So it's not an unusual use case even for people who don't wear glasses every
Yeah.
day. So then the next step after the glasses, what could that be? Well, that's obvious. It's the combination with your brain.
Yes.
So that's brain computer interfaces, BCI. And BCI is a rapidly evolving field, not just Neuralink, which has made some very impressive progress, but also Science Inc., which I'm invested in. It's a very exciting company that actually puts an overlay here into the iris and allows people who can otherwise not see to suddenly see again by connecting directly to the nerves. So it's a very, very exciting field that will help people initially who are in some form limited physically, but in the future it will connect to everybody who wants it, and
Yep.
basically gives you an extra. Imagine if you could surf the web, and instead of always finding the next thing or giving a prompt, what if you could just think, and the next thing
Totally.
shows up
Yes.
immediately that is interesting to you just by thinking about it. So
Now
that's that.
we talked about this novel interface so many times, right? Like we had MIT lab trying to do this with the hand movements, all too complicated, right? Also, the AI tool where you then double tap on your hands doesn't really make sense. But like to have a brain connection will be definitely the next helpful interface.
Right. And some people will say that's a disaster. I never want that. And of course, I understand that. Look, there are two different ways of thinking about this. One is the invasive way, which is the world that we're going to live in for the next 20 years, because it is very difficult otherwise. But then there will be probably some breakthroughs that allows us to be non-invasive and actually just read what's in our brain.
Yes,
And then, of course, if you want to go even further out, if you can read, but you cannot write, that's a big limitation. What if one day you could actually write thoughts into our brains? So now you're talking about Big Brother and what are we supposed to think and how forced to think? And there are all kinds of problems that come with that. And I want to be also clear about that. Also, when we talk about biology, this is all exciting and amazing, but it has to be handled with great care.
totally.
And, and,
With
and the reason I'm saying that is it has always been like that. Whenever we, as humanity, develop new ideas, new products, new technologies, it was always both a good thing and a dangerous thing.
yes,
For example, in the past, you had horses on the street, and then cars came, and one day the first person died from a car. And you could say, well, that's a disaster. It's terrible. So now, should we stop having cars because of that? No, but you have to develop the safety standards around it. But the difference is with every new generation of technology that we're developing, the stakes get higher.
totally. Because the scale gets higher.
The
Now,
scale gets higher.
before we dig into this, because I obviously want to talk about early. You have a nice swing
Yep.
in the background. We should cover this. But if the media team could just play again the question thing in, because as usual, you can ask questions. You can ask questions about scale. You can ask questions about whatever you want to ask questions about. We will see it. We will bring it in here.
Mhm.
let's dig into the topic of today, which is essentially the whole idea of bio-AI and AI interfaces to the human. Tell us a little bit about Early and tell us about how you met this initial idea, because that's super fascinating story.
All right. So first, the one big area that you asked about big areas, physical AI. One of the most fundamental ones is the living atom meeting the digital bit, and that is your body. And that's where we are in biology. And that is precisely why I'm doing what I'm doing. Because I think one of the most foundational changes that we're going to see in the next 20, 30 years is the combination of the living atom with the digital bit. So here's what happened. I was looking for my next thing after my last company got acquired, and I was looking at over 200 ideas. And whenever I look at a new idea, my first filter is to try to forget it as fast as I can. And most ideas don't pass that filter. They're forgotten very quickly. And only those ideas that I simply can't forget despite trying, might be interesting. So in general, the underlying theory is that I generally believe there's an ocean of good ideas, but there's a dearth of outstanding ones. And only outstanding ones are worth pursuing at all. By the way, not even every outstanding idea is worth pursuing. So you have to really have a very tight filter. So then this is what happened. I looked at all these different things. I already mentioned brain-computer interfaces. I mentioned consumer robots. And then I was really excited about biology meeting software. And I only live 4 miles from Stanford in a little town called Portola Valley, 4,000 people. And so I rolled down the hill and went to Stanford and stumbled around the campus. And I met really interesting people. And it's interesting. I don't know how many people I met, probably over 100. And each person was telling me they were working on the world's most important idea. And by definition, everyone except one or everyone except everybody was wrong about that, of course. But the problem was I'm not an expert in biology, so I couldn't differentiate between a good and an outstanding idea. I could perhaps say whether something is good or not very good, but not whether it's outstanding. For that, you really need to know your stuff. And so it became really frustrating. And so, like after the first 4 months of not doing anything were really exciting, went to China and packed up our little micro children, and my wife and I went there and we lived in Beijing for 3 weeks and met entrepreneurs from tiny idea to big idea. Went back, went to Europe, did the same thing. Wonderful. After 6 months, I became very, very antsy. I wanted to do the next thing, but I couldn't find it. And so now, mind you, 11 months in, I was really depressed. It was like now Thanksgiving, and Thanksgiving is basically the end of the year in the US. And I was sitting there thinking, I still don't have it. I can't believe it. The year has passed. And on that day, the door opens and my wife comes in, Angel, and we lived in somebody else's house as a sublessee at the time because our own house was being remodeled. So we got other people's magazines that were not meant for us. And she comes in and she gives me this magazine from Stanford Medical School. She says, I think you have to read this story. So I look at it and it's called And Yet You Try. You should Google it. It's a
Oh, wow.
story in the Stanford Medical Magazine. And
Okay.
in the story, it explains how one of the world's most preeminent early cancer detection and treatment experts, Dr. Sam Gambier at Stanford University, lost his only child to cancer at age 16,
Yes.
Millen, and how he was searching for new ways to detect and treat cancer early, which is obviously viscerally important to him. So I read this story, Lutz, and I was
Can you
shaken by it.
can you like the and it is in your name early, like you said, like he was researching how to detect it early.
Yes.
Many of like many folks who are not from the medical field might kind of think, well, what we are looking for is a treatment. But early is about detection early. And the guy, like the professor, was searching for ways to detect it early. Why? That's a slightly shift of notion here. Why is that?
Well, the biggest lever is to treat your cancer early. I give you an example, lung cancer. Lung cancer is the number one killer of all cancers. It kills twice as many people as the second biggest one, which is colorectal. And it's not just smokers, by the way. So lung cancer, if you detect it at stage 1, you have over 60% survival rate after 5 years. If you detect it at stage 4, you have an 8% chance of living in 5 years from now. 92% will die.
Yes.
So it is absolutely critical to treat it early. You can only treat it early if you diagnose it early. But to be clear, Earli started as a diagnostics company. It's now a therapeutics company. And we can talk about this in a bit. Do you want me to explain how this thing came about?
Yes. So you actually met this professor, right?
So I decided in that moment on this Thanksgiving Day to send a cold email. So I sent a note to Sam and I said, it is not a coincidence I'm sending you this email on Thanksgiving Day. I can only imagine how hard this day is for you. I don't even know your son, but I felt a gripping sense of grief when I read your story. But I was also very inspired by the work you're doing. I'm not a biologist. I'm not a scientist. I'm a serial entrepreneur and I'm looking to build my next thing. I would like to meet you. It took 2 months to find a spot on his calendar. And on a Saturday morning, we met for breakfast at a small restaurant in Portola Valley. And the first thing we found out is he also lived in Portola Valley.
Oh, fun.
There are
Yes.
only 4,000 people, and we were neighbors and didn't know that. It takes 3 minutes by car from one house to the next. So, my first question to Sam, and might be relevant for the audience because I am not a biologist, I'm not a scientist. What the heck am I doing here? And I said to him, I need to ask you something, and I'm not fishing for compliments. I want a brutally honest answer. Should someone with my background, or better, lack thereof, because I'm an engineer, not a biologist, bother the world of biology with my presence? And he looked at me and he said, I have two answers for you. The first answer is the world of biology needs more people like you, not fewer people like you, because we have a lot of experts and very few generalists. We have even fewer people who know how to build a startup. And the second answer is the world of biology will always find a way to screw you over. It will take longer than you want, and it will be harder than you want. So I took the second part of the answer as the cost of doing business, didn't like it, but went with it. And before I knew it, every other Saturday morning, I found myself sitting at his kitchen table at his own house, and he was trying to teach me biology, largely in vain, but I give him credit for trying it. Then he started introducing me to a bunch of awesome scientists at Stanford and clinicians. The problem was I still couldn't find the right combination of an outstanding idea and an outstanding scientist or teammate or possible co-founder because each one is 1 in 10,000. You can do the math.
Yes,
You
totally.
multiply them. So 3 months later, I went back to Sam and I said, Sam, I'm so sorry. I still don't have it. May I ask you a simple question? You've seen hundreds of ideas in your own lab, outside of your own lab. Which one has the highest potential of all of them? And he looked at me in his scientist underselling type of way. And he said, there is this one idea that I believe has some real potential. And that became Earli.
Now, let's explain. By the way, we have again, like audience, please ask questions. I have already six of you, like six questions here from the audience, which is amazing. But looking at the questions, folks understand very much what Early is doing. At least the ones who asked. Thanks so much for it. Now, give us first the spiel. What is Early? What are you guys doing?
All right,
Um,
I'm going to keep this brief. So, very high level. The number one problem with cancer drugs is what? It's systemic toxicity.
you kill like cancer are cells which the body didn't identify as bad cells, and you need to kill cells, and you just need kill cells. With radio, you kill not only the cancer cells, you kill everything around it. With chemo, you're not only killing everything around it, you just kill everything. So yes, if you don't have a biomarker like a CD20 marker or something like to actually attack it directly on the cell, you are screwed.
That's exactly it. So, the top 10 cancer drugs produce 87 billion in revenue per year. And these top 10 cancer drugs, which are very potent drugs, give you on average 12 months more to live, and they give you 15 percentage points more survival rate, which leads you to 49% in total, which means every second person still dies. So why is that the case? The main problem is when you dose someone, the drug doesn't only work on the tumor, it also works on healthy cells. And that's, by the way, why in chemotherapy your hair falls out. The hair doesn't have cancer. The chemo drug is focused on fast dividing cells, and your cancer divides fast, but so do your hair cells, of course, because your hair is growing.
Yes.
That's why your hair is falling out. So, in other words, extraordinarily imprecise.
And by
And
the way,
if
this is why one of the first questions we got here in the chat was from Rabab. Thanks for asking, by the way. How do you avoid that this is leaking into benign tissue, right? How do you
he,
exactly that?
he just asked the $1 million question. So, I'm going to try to get there really quick, okay? So, let me just explain briefly so you understand the size of the problem. Now, if you give somebody a drug and it has these so-called on-target but off-tumor effects, in other words, your target is not only on the tumor, it's also on healthy cells, then you only have one chance of addressing that. What do? You
Yeah,
lower the dose. And when you lower the dose, you lower the efficacy of the drug. So you're not giving the person as much as it should. And then the second double whammy comes in. The drug goes into your bloodstream. You have 5 liters of blood in your body. By the time it reaches your tumor, there is this much left. So now I'm going to ask you a simple question. The question we asked ourselves. What would be the ideal cancer drug?
the
How would
ideal
that look like?
that is so far, before I got to know you, so far it was understand whether the cancer cell has something you can dock on, like
Mhm.
CD20 marker, it's a typical thing. So it has some structure which identifies the cell as a cancer cell, and
Right.
then you create a drug only focused on that genetic structure of the cancer
Yeah.
cell. The only
Yeah.
problem is most cancers don't have that structure where you
That's
can
right.
dock on.
They either don't have it or it mutates a little bit from patient to patient. So it doesn't work everywhere. And then it, unfortunately, not only on the tumor, it's also elsewhere. And that creates the so-called on-target off-tumor toxicities, which lowers the dose. We're back to the same problem. So
Yeah.
now let me ask you a simple question. What if we could make the drug only where it's needed, meaning in the tumor, have it be produced by the tumor against the tumor. In the tumor, by the tumor, against the tumor. What would be possible if we could do that?
Well,
It would
it can identify the cancer cell, make the cancer cell destroy itself, right? I mean, that would be personalized cancer treatment on the level of one.
yeah, so it would essentially unlock extremely potent therapies that would otherwise be too potent because they also go elsewhere and they're too toxic.
Yes.
So we're going to try exactly that in a lung cancer phase 1 trial starting next summer, dosing the first patients. So how does this work? In order to make this possible, we had to create three major innovations. We created the first genetic switches made out of DNA that only turn on in cancer cells when you inject them into the body. They go everywhere, but they only flip on in a cancer cell, like a light switch. And that required massive amounts of machine learning and AI. Otherwise, we wouldn't be here. In fact, the interesting thing is we tried to do it without AI and it failed. I'll get to that in a moment. The second big thing is somehow you got to get this DNA thing to the tumor. How the heck are you going to get it there? And that's where lipid nanoparticles come in. You might have never heard of a lipid nanoparticle, but actually, most likely you've had it in your body already with the COVID vaccine, because that's how they became very well known and billions of people have received them. Here's the problem with them. They are excellent cargo containers that take DNA or mRNA and they transport it through the body. The problem is, in 10 minutes, they're in your liver and they're out of your body. Why the liver? Because the liver is responsible for flushing everything out of your body that doesn't belong there, from alcohol to lipid nanoparticles. And so therefore, if you want to treat any cancer but liver cancer, you're going to have a completely different lipid nanoparticle. And for 15 years, this has been called the billion-dollar barrier because it's exceptionally difficult. This is chemistry, not biology, by the way.
Yes, and I mean, these are two things, right? First, like you need to reprogram the cell, and the other one is you need to do it in the body at the right place.
That's right. So first you got to get the material there. That's the lipid nanoparticle. So you need like a cancer lipid nanoparticle that gets to the cancer and into the cancer. Once you're there, you got to make sure you have a switch that only turns on in cancer, because the lipid nanoparticle is also going to go elsewhere, not just to your tumor. Now, that switch has to be exceptionally accurate.
So how do you do this?
And then the third innovation is once you are actually in the cell and the switch turns on, you have to program the cell to make its own drug. And since you're only getting into 1% of the cancer cells with this transfection, with these lipid nanoparticles, the big question is, how in the world are we going to kill the other 99% that we don't get into? And the answer to that question is, think of a domino chain reaction. If you could take the 1% you get into to make a drug that activates your immune system to then come and attack the whole cancer and kill it, then you would have that done. So
but that means the drug needs to make the cell identifiable. And that means we as humans, as we took the cancer out, we weren't able to find something which is identifiable. And now we rely on the immune system to figure out something which is identifiable.
That's exactly right. So the cancer cell needs to produce a message. Think of it as a smoke signal that comes right out of the tumor and then have the immune system attack the cancer. But you have to be very careful that what you're doing is staying only in the tumor so that you don't have these off-target effects elsewhere in the body, these side effects that you don't want. It has to be exceptionally local. And all that brings us back to the switches that you asked about.
So how do you, I have two questions. One is in order to make clear, how do you do this? And the other one is actually one of the audience questions here is from K Carr. He said, okay, this is all cool, but how do you communicate this? Because that's complicated, right? How do you educate and influence all the decision makers in the chain? Because what we are now discussing is novel. It's a novel way of doing therapeutics. So far, we could not reprogram the cell. So far, we could not get it down to the level of one. So far, we could not have the cell producing their own drug. They all need a license. Yeah, every cell needs now an FDA approval license here. Think about it. No, but joking. But still, tell us how.
The short answer is very simple. We're killing tumors. And that's, at the end of the day, the only thing that truly matters. It's like, would you like to make the drug only where it's needed and then kill the tumor, but keep it out of the body of the rest of the body? And the answer is obviously yes. And then the question comes, well, how are you doing this? And then I can explain Well, it required three innovations and they took years to accomplish. This is not something that you can, yeah, it's easy to be said, but it's exceptionally hard to do. So it took us years to do it. But, in the beginning, you find some people like Mark Andreessen, Jorge Conde at Andreessen Horowitz, Mark Benioff, who literally signed on to the concept itself because Sam Gambier was brilliant and had shown at Stanford with very early mouse models that there might be an idea here. And they signed on to that. And kudos to them for taking the leap of faith with a $20 million seed round on that. When we then went in the A round and we brought it to Khosla, they were betting on this at a time when we just had some early proof of concept in mice, but not a lot of data yet. And they led a $40 million round with Perceptive. And then afterwards, we started going to town and we had to use tons of machine learning, 20,000 cancer samples, sift through them with machine learning because the original concept was to use a switch that nature provides. It's called an endogenous promoter. It looked really good in cell lines that never change in the lab. But the moment we tried it on individual real cancer patient samples from the operating room, the whole thing failed completely. We literally hit a brick wall. It was a lot of brick.
A lot of great. And how do you, because like, you have been entrepreneur for so many times. You said it by yourself. You need three miracles. Most startups fail on one miracle, and you actually have now three in a chain. How did you go about the brick, and how did you
Well,
make this feasible?
I hope nobody was aware it was three bricks. I didn't even know that it was. I knew the major risks. That's one of my main thing when I look at an idea. I talk to experts, not for the inspiration, because experts are really good at telling you why something will not work. So they're very good for hole drilling. So I knew the kinds of risks. Delivery was going to be the major problem, getting enough to the cancer cell.
Yeah.
And then afterwards, the specificity, meaning is it cancer or not? And then the sensitivity, meaning are you detecting it across all patients that have cancer or only in some of them? So those were the major three main risks in order of priority: delivery risk, specificity, sensitivity. And that has never changed. So the analysis is accurate even so many years later. So now, to your question, how do we solve it? We actually said, look, we could look at individual mutations, like lung cancer has over 100 of them. The problem with that is once you do that, your construct gets bigger and bigger and bigger to check out all these mutations, and you can't get it into the nucleus anymore of the cancer cell, which is where the dysregulation happens that causes the disease. So you're stuck between a rock and a hard place. Either you're accurate enough, but you can't get into the nucleus, or you can get into the nucleus, but you're not accurate enough. So what are we going to do about it? And then it took us a year of failed attempts until we asked the right question. As you know, oftentimes finding the solution is mostly dependent on asking the right
Yeah,
question.
totally.
So we said, if we cannot interrogate every single mutation, what if we didn't interrogate any of them? What if,
Interesting.
what if instead we looked at the downstream commonalities of these dysregulations, the things that are caused by them, not the original mutation, and looked at the so-called hallmarks of cancer, which Hanahan and Weinberg described 20 years ago, such as rapid cell proliferation, lack of cell death, and so on. And we started interrogating the results and not the causes. And by doing so, we drastically narrowed the scope of the problem. We then went through 20,000 cancer samples and so much data that 4 of the largest AWS Amazon instances ran for an entire month just to get through the data a single time. Once we had done that, we had a shorter list and we identified so-called master transcription factor binding sites that are dysregulated. We then assembled a combination of those, but then the real work began. And this is where it gets really tricky. So let's say you find the dysregulation of these transcription factors. You can now put them together, but you can space them, direct them differently. And it's a three-dimensional system. So there are literally trillions of possible combinations, especially when you add a core promoter like we did and an amplifier and so on. So you have such a large universe of genetic sequences, you can't get through it.
Now, I love this structure you just described. And by the way, we probably want to level it up a little bit more in terms of the technology description.
Okay.
But if I just point to the flow that we are having, very often people say, oh, generative AI, they will solve all our problems and we will have those amazing new drugs. Listen to Cyriac here. Yes, he described very well how he uses strong machine learning power and large clusters. But as he said, the real work really starts once you have the understanding.
Mm.
Because the machine in a BioAI in the interface is not solving for the application. It is giving opportunities to figure out this would be one way to address it. So there are many roads to go down, and the machine helps us to go down those roads faster. And that's the real value when we talk about AI. Now, fast forward, like you actually figured out a way, and then you went, because I have here one question from Chris about like how to apply this leukemia in children and so on. I was like, this is early days. Cyriac is not yet there. However, you started with dogs, right?
Yeah, we actually have a simple North Star early. We want to be in patients as fast as we can. So we're taking our construct and we want to be in humans by summer of next year.
Summer of next year. Amazing.
So we actually want to run a clinical trial on lung cancer patients, a phase 1 trial, where we will intravenously inject the compound and we will try to show that this is safe and show some early efficacy data or indicators that point to that this can actually cause effect on the tumor as we are intending it to do. Because the goal of this thing is not to be a theoretical research project in a lab forever. The goal is to save people's lives as fast as we can. And I should probably point out that Sam Gambier that we spoke about died from cancer 2 years into the company. He had a cancer of unknown primary, which is even worse because you can't even detect it early. By definition, it was stage 4. And it was very sad. And my other co-founder, David Sui, who's a brilliant gene therapy expert, and I have built this together with Badri Anantharayanan, who's our SVP platform, into the company it is now, based on the original vision, but in a completely different implementation. We haven't even gotten to LLMs yet, which are also part of our game now, because the wet lab, this is something I should probably say, because I think it's really important for this topic
Yes.
of
Tell us.
physical AI. Physical AI is only interesting when you train it on physical world data.
Yes. By the way, this is a general problem for the whole space. I get asked very often what is at the moment the best startup idea. And obviously, I don't know because I'm not a wizard who can look into the future. What I do know is that all models need real data and therefore physical data and like the physical world models. Jan LeCun and so on talks much about it. They need data and they need real world data.
That's right. They need real world data. So what you in biology need to do to get real world data is to have a wet lab with pipettes and,
Yes.
and a vivarium with mice. And you need to get all this data generated in actual wet lab situations and generate high quality data, because otherwise you've got garbage in, garbage out. Everybody knows that. So how do you generate that is the problem. Because normally when you do a sequencing or a test on a sequence, you can do, like, if you're really, really intensely working, 50 a week.
Yes.
Well, unfortunately, we had millions to test. So what are we going to do? Be there for 100 years? So we hired an expert from Austin University of Texas. She came in and she started building a so-called massively parallel reporter assay, which allowed us to look not at 50 a week, but to look at 250,000 per batch. By genetically barcoding them, like a product in the store that you get checked out at the register, each one has a barcode, except it's not a black and white code. It's a genetic sequence. It's a DNA sequence. So we throw them all in the same pot. And the ones that are winners, that are total outliers, that are way better than others, we then read them out based on the DNA barcode. And that data, the best ones, get fed into an LLM. Why an LLM? Well, because an
Transformer
LLM,
structure, let me guess.
what is DNA? It's text.
The sequence. So it's
It's
a
a
sequence
it's a
completion.
it's a sequence of text. And this alphabet of DNA has 4 letters, and they keep permutating over and over again. So there's a perfect problem opportunity to be solved by an LLM if it's trained on the right data.
And
So we
this, hold
started
on,
with
this
an
like we need to make this clear that people don't get confused. When Cyriac means LLM, he means like a technical structure, which is a transformer architecture, which
yes.
is essentially trained on sequence pieces. The
Mhm.
LLM you guys have in Anthropic, Claude, OpenAI, whatever tool you use in a chatbot is trained on human data. So its life is like a box of, next word is chocolate, because it's trained on it. If you look at a sequence of
Yes.
um, gene sequencing and you are
Mm.
trying to understand what's the next possible mutation, you
Right.
have the same thing it's like a box of chocolate. Or if you look at fraud data and saying like these are transactions, what is the next transaction to be? We use large language models as well, like we use transformer models for those types of problems.
That's a great way of explaining it, Lutz. Essentially, we're predicting the next possible sequence for a genetic switch.
Yes.
And here's the interesting thing. All the models out there, they're okay-ish for DNA. There's one that's specifically trained on DNA, which is Evo2 by the Arc Institute. And that's actually not one of the models that everybody uses as a chatbot today. It's the best one that's out there right now. But still, 99% will be useless stuff that comes out. So how do you predict what is probably not useless stuff? And for that, we build a proprietary oracle, a prediction model that is much better than market standard to predict what comes out. And that what comes out, we then use bioinformatics to decluster it, to make sure it's not so concentrated that they're all pretty much very similar. We spread it out, diversity gets bigger, and then we feed that data into our wet lab. The wet lab
Yeah.
then produces the outliers. The outliers go back into the LLM. And so with every spin of the wheel, this thing gets smarter. It's a learning loop. And so the last thing I want to,
Let me like there's so much amazing stuff is happening here. Like
yeah,
I want to underline this because this is very, very cool. Many folks saying, okay, and OpenAI launches now yet another model. What is out there for us as an entrepreneur? Or will like Google and OpenAI take over the world? And you just heard the very clear explanation of why this is not so easily feasible. Because Cyriac uses a transformer, the one which is the basis for OpenAI as well. However, he uses his personal data, not his personal data, but the data from
the
his
wet lab
lab,
data.
from the wet lab data, which nobody else has, to
Right.
train a fine-tuned model that can answer information that nobody else has, and then he does a feedback loop to improve it. And we have seen this so many times. The future at the moment is really on so-called fine-tuned models that are trained on specific data nobody else has. And therefore, there is a race on data, there's a race on fine-tuned model. And the example Syria just gave is for healthcare, and it's very specific, but the idea, the underlying idea, is happening in all industries across the globe.
Yeah, you probably remember what AlphaFold did
Oh,
for
yes.
proteins. AlphaFold made proteins programmable. So what do? We are essentially making DNA programmable.
Totally. Yes. And therefore, you have the ability to do this feedback, which is stunning. Very,
And
very cool.
the oracle turns out is one of the most important elements. Having a prediction model that is proprietary and better than what's out there is really important because otherwise you have too much garbage that you can't use. And then the wet lab is the one that qualifies it again. And then it's the learning loop. You need learning loops in order to
Yes.
accelerate the progress. So that's what we do. And that's essentially where we are now. We've tried this in thousands of mice to kill tumors in various mouse models. We have shown safety in monkeys, which is exciting. That just happened in the last 4 months.
Oh, wow.
Now we can show that it's safe in monkeys. It was so safe that the body temperature didn't even go up, which is really remarkable for us. And then now we have to we lock down our clinical candidate, which means the product we want to do, and now we want to take it through the FDA and manufacturing process. This has already started. We have a very strong CMC manufacturing team on board, and now we're taking this to the clinic.
Now, because we are like an AI engaged audience here, we have certain questions which are interesting in a healthcare setup on here. So Vina, thanks Vina for your question. She actually asked about liability. She says, okay, well, if you now do an AI design genetic switch,
Mhm.
and let's say, like, you produce false positive, false negative, because it's, that's in any system the fact. How do you actually treat this in terms of liability and responsibility? And it's interesting because it's a question you have across all AI design products,
Mm.
but healthcare is completely different in a certain way, which makes it more complicated or less complicated. It depends on which
Yeah.
setting.
So, healthcare is different in good and in sometimes not so good ways. As I mentioned before, even the best cancer drugs oftentimes, unfortunately, don't work today. This is the reality we live in today. I'll give you an example. You know what checkpoint inhibitor drugs are?
Yeah, I do, but I explain the checkpoint
So
inhibitor
checkpoint
drug.
checkpoint inhibitor drugs are the biggest innovation in cancer treatment in the last 20 years. They essentially use your immune system to attack the cancer. The
Yes.
way they do it, and the big insight came from Jim Allison. Jim Allison basically found out that you don't just need to stimulate the immune system to do more. You need to first take the foot off the brake of the immune system. That means the cancer is really good at telling your your immune system actively not to intervene, not to attack the cancer. He found a way to take that foot off the brake of the immune system, and he got the Nobel Prize for it in 2018. And all of these checkpoint inhibitor drugs make up over half of the top 10, over 40 billion, 50 billion a year in these top 10 cancer drugs. Now, here's the problem. Even the best drugs like that have on average a 35% response rate. 65% don't respond to it.
Yes.
And the
Because
reason
again,
is,
you're going on such a meta level in that
yes.
you're missing the the molecular structure threat.
Now, the biggest problem is that if you take the foot off the brake of your car, but then you don't push the gas pedal, where does your car go? Not
Slower.
far.
Yeah.
It stands still. So you also need to stimulate the immune system. But when you stimulate and you push the gas, you get these cytokine storms throughout your body. That's the problem. That's precisely what Early is doing. We want to be the second piece of the missing puzzle, the second missing piece of the puzzle. Somebody takes the foot off the brake, but somebody needs to push the gas, and we do it locally on the other tumor. So the reason I'm using this as an example is because our audience member asked us about liability. The biggest problem is that a lot of drugs do not work today in general. So if you can get from 35 to 70%, you've just doubled the rate at
Yes.
which these drugs work. So now you have to be safe. And that's where a lot of mouse tests come in, a lot of animal tests, monkey tests, all kinds of safety tests before you introduce a new drug to the market. That's where the FDA comes in to make sure that you are taking the right approaches. And safety is first for any new drug. And
By
every
the way,
single
for
drug gets
me,
tested this way.
like it's a given that from everything which we deploy has false positive and false negatives in any industry, right? And what I learned at my time at Google Health by working with the authorities, actually, no area in our life is so well thought through like healthcare in terms of when do you start screening? How long do you start screening? What does it mean for population health setups? No, we think about healthcare in this respect so much
It's
in
very
terms of
strict.
very
It's very
strict.
strict. And it needs
So
to be for safety of patients.
yes, so this is actually like Peter had another question. He talked about what happens if you, during those tests, discover conditions that you haven't expected. It's the same thing, right? You're trying to kill cancer. If you're realizing that there are side effects, then it becomes a very strict protocol, which makes
Absolutely.
healthcare one of the best AI regulated areas, actually.
That's right. I mean, that's why clinical trials are there. And I also need to be clear, our genetic switches are not all machine designed. We are constantly giving our own input and modifications to it. This is a combination of humans and software,
Yeah.
humans and machines. So the biological knowledge is the critical factor here. You don't just come up with random stuff because by that, honestly, if you try that, that's called brute force. You'll be here in 100 years from now. It won't work.
Yes. Now I have one other question because you are going now towards a clinical trial. You narrowed the cancer you're trying to attack brutally
Yes.
from there. So many cancers you could have used to attack, and you narrowed it down to one area which you're now going to clinical trial with. This is a little bit a question every executive in this country has because AI can do so many things and they need to figure out how do I deploy. So if you think outside of healthcare for a second,
Yes.
as a as the executive Cyriac who you are in terms of how do you have focus AI resources, how did you decide on the actual trial structure where you wanted to go? What was
Yeah.
for you the driver?
You really have to get your hands dirty and understand at a very fundamental level where the main problem lies. And for that, you need to really dig into the business you're in. The business is the wrong word. In this case, it's about patients' lives. Where is the problem in the clinic? Where do the patients suffer? Where do the drugs not work? Where are the problems that are limiting? Why are they limited? And the deeper you dig into that, the more insights you will generate where it fits. And to be clear, it's also important to say this. You don't start with the tool, you start with the problem, and then it will naturally emerge which tools are best to use. Like try to imagine you want to build a house. You want to have a house, you want to have a roof over your head, and you want to feel really comfy. And now you're going to start planning the house. What do you want to have? What's the output? Now naturally you're going to find the right tools to build that thing, right? And you don't start with a screwdriver and say, what could I build with that? You
Exactly.
start you start with, hey, I really would like to have a house that's really beautiful. Let me design one. Okay, so now I need some materials, I need some tools, and now I'm going to get to work and I need some skills. I need really skilled people who know what they're doing so that the house doesn't fall on me when it's finished. So all of these same principles apply. This is all very, very normal, rational, standard thinking. It just put your own brain to work and it will automatically emerge.
Which is, and I think like it's actually a very nice summary from
Common
all
sense
like
is what I mean.
you say this slightly, but like if when I talk to many executives and I talk to many boards, and the question is how do we deal with AI? Now, if I just tell them common sense, Cyriac says common sense, then it's
Yeah,
very
I don't want
hard to
to.
make
No, no, I don't.
action.
I want to be clear. I don't want to come across like condescending in any form.
No, no,
It's
no. It's
it's
like it is
it's far
common
from what I
sense.
what I what I'm trying to say is let it naturally emerge from the
Exactly.
from the problem because you're facing certain problems. And now all of a sudden you've got this really powerful tool that you didn't have before in your toolbox. Now you
Exactly.
can apply that tool because it's so good at so many things that you weren't able to solve before.
Meaning the core part here is actually, and you said it very nicely, Cyriac, understand the problem space. So when I just recently, like as many as of you, like of many of the listeners may know that I publish at Forbes, right? So I wrote this article about what do boardroom members need to know about AI. And essentially, it reads like common sense because what I say, the underlying focus doesn't change. You focus on your business, you focus on your patients, you focus on the problem at hand. What
That's right.
has changed is the tools, and you need to understand the tools.
Yeah,
And
it's
therefore,
basically all of a sudden you've got a bazooka in your toolbox.
totally.
You've
But you
got this
still need
in
to aim. You still need to pull the
you
trigger.
need to
You
you
still
need to
need to
know
load.
exactly what to do with it. But there's this really powerful, smart tool sitting there that you didn't have before, and that's the change.
I could spend more hours with you, and I should actually do. We should have another coffee at one point in time. But Cyriac, thank you so much for being here. Like to all the people those questions I did not ask, I'm very sorry for it, but we got about 50% of the questions done. Cyriac, it was a pleasure to have you here. Thank you so much.
It was really fun. Thank you so much, Lutz, and hope everybody got a little bit out of it. Appreciate it.